It's fine. As long as we don't get kicked out, it doesn't matter.
Hello.
Hello.
Right. Everyone's here. Oh, hang on. Just 1 second. Looks like no, it's public. Weird. YouTube told me we're private. But apparently we're not, so that is good. Okay. Now everyone's here. Chris, you're presenting. I'm going to make you co-host so you can share. And other than that, welcome everyone to Neuroccino on the 27th of November. Just so I don't forget, at the end, we have a CNS talk this week on, well, by Nicola on Wednesday, 4 p.m. And she's going to talk about neurotransmitter receptor distributions and what that can tell us about the structural functional segregation of the brain. So do join us for that Wednesday, 4 p.m. And other than that, I'm happy to hand over to Chris to talk about non-invasive deep brain stimulation. And he already shared the paper in the chat. Thanks for that. Chris, take it away.
All right. Is the size of the screen all right? Yeah. Right.
Yes, perfect.
Okay, so I saw this paper on Reddit, on the science subreddit, and I thought it was interesting. That's not really my domain. I'm not working in stimulation. I'm just working with the data. But I'm interested in the different techniques that can maybe allow us to do new things that were not available before. So this one is about non-invasive temporal interference electrical stimulation of the human hippocampus. So first they explain that deep brain stimulation has been used to help patients with multiple diseases like Parkinson's disease, obsessive compulsive disorders, affective disorders, and even there are some experiments to see if it can help with Alzheimer's disease or depression and other disorders. But of course, the problem of deep brain stimulation for now is that you need surgery, you need to implant electrodes to stimulate deeper structures in the brain. And there are some non-invasive methods that exist, like transcranial magnetic or electric stimulation, but they have some drawbacks. So first, they only reach a certain depth in the brain, but also their stimulation tends to leak in the area around the area that we are targeting. So it can have some unwanted side effects. And in this article, they present a method where, so I'm going to show the first here, and especially the first part, ABC, where they use a similar methods, or they just use electrodes on the outside of the brain, so it's non-invasive. You don't need surgery or anything. But in that case, they use two sets of electrodes to send high frequency electric current in the kilohertz that will only interfere at the sites that they are targeting. So when they interfere, the frequency, they can modulate the interference, and the frequency is going to reduce, and they can also modulate to have the amplitude that they need for the stimulation effects that they want. And the advantage of that is that it is, they show in the paper that it is quite localized. So first they show it in a simulation, and they show that if we compute the currents that we would have, they have a software that is called MIDA, that can take into account the tissues resistivity and all the electric properties, and they calculate that you would actually have the location of the interference, so where the stimulation would be, as they can expect. But of course, simulation is not enough. So then they try it in a cadaver model, where they implant the cadaver with actual electrodes with probes to see to measure the currents, and here they target the hippocampus, and they show in the cadaver that the stimulation location is quite focal. They also show that they can modulate the current. So they can, for example, if they increase the current in one electrode, it can shift the location of the stimulation. So they can even target subportions of the hippocampus. So I think the locations are a little bit crude, but it's still pretty impressive. They can target the posterior or the middle or the anterior part of the hippocampus. But for now, they just showed that in a cadaver. Also, they explained the limitations, that the cadaver doesn't have the same temperature as a normal human. So they need to test it in humans. Of course, this method has been tested on animals before, and they have all the ethics to be allowed to do that, of course. And they tried it on 20 healthy participants, to see if they could. So first they used a current that was just enough to change the bolt signal in fMRI. So they put the participants in the fMRI and stimulated them with the technique, and they showed that they actually have a different fMRI activity, a different bolt signal activity, and that they can even modulate the current to change the bolt signal in a part of the hippocampus and not the others. And with this experiment, they also show that only the targeted region was actually stimulated, and the bolt signal in the surrounding areas was not significantly affected. But they also show that there is a stimulation in changes in bolt signal in functional networks. So they talk about an anterotemporal network and a temporal medial network that are connected to the hippocampus, and they show you a difference in the bolt signal with these regions. But of course, at this point, I forgot to say, these participants were doing a task that typically involves the hippocampus. So it's a face name association task. And in that case, they only affected the bold signal, but it didn't affect the performance, because they explain that they need to show that, because affecting the performance would be a confounding factor, because if there was a difference of performance, they would also they couldn't disentangle if it was a difference of bold signal because there was a difference of performance, or if it was because of the stimulation. So first, they just showed a difference in bold signal, and then they used another cohort of 22 healthy participants to actually explore the effects of the stimulation on the episodic memory of the performance in episodic memory. So they use the same task. If they show it in, yes, they have another figure, and they show that they can actually improve the recall performance in the face name task. So in the second experiment, they changed the modulation of the stimulation, so the stimulation is stronger, but also they do it for a little bit longer. I think it's 30 minutes. I don't remember if it's in several parts. I think it's like 30 minutes of stimulation. And they show that the stimulation actually improves the performance in episodic memory tasks. And then they talk about if there are some adverse effects, and they didn't record any in the two cohorts that they tested the method in. There was only mild side effects, like an itching sensation on the electrodes, on the electrode sites for some participants, but they say that it's pretty common with the stimulation. So in summary, the article shows that their method, temporal interference, is able to specifically target deep brain structures, and it can even modulate the current to stimulate subportions of the structures. They show that the stimulation can affect the bold signal and the functional connectivity without affecting the behavioral performance significantly if they want. And if they modulate the current, they can significantly affect the performance in a memory task by stimulating the hippocampus. And they show that the method is safe and tolerable. In the limitations, the authors say that with this study, it doesn't directly allow them to disambiguate whether the bold signal change was due to vascular changes, or actually by neuronal activity that the stimulation changed. But they say that it is quite unlikely that it is only vascular, because the regions around the stimulated region were not affected. And also, I don't think they say that, but if it was only vascular, I'm not sure the functional connectivity would be affected, but I don't know. I don't know if that's correct. But that's just the thought I had when I read the limitations. And of course, there is another limitation. They use two quite small cohorts of participants, so 20 participants and 22. So of course, to generalize the results, it would require bigger studies. And also, we can't use their study to generalize the brain behavior relationship. It's only to show that the stimulation actually has an effect but it's not to show the to generalize the episodic memory performance and stuff like that. And in the end, the authors, they explain that this method has great potential to non-invasively help patients with different disorders. As I said, like potentially Parkinson's disease, compulsive disorders, and things like that, and potentially with other disorders like Alzheimer. But I think it's also interesting to have a method that can pretty much target any structure in the brain safely without having to use surgery. And potentially there will be some cool experiments like there's been with transcranial magnetic or electric stimulation. So that's why I was excited with this new technique. So I think I went a little bit fast, but that's not super my domain. So I also wanted to discuss if people think that it could be a method that could be used in the future for experiments. I don't know. I participated to some experiments with magnetic stimulation, and I didn't do the study myself. I was a participant, but I thought it was really interesting what you can do with that, and if you can actually target any structure in the brain, that could be really cool. So yeah, I don't know what you guys think about this research.
Thanks, Chris. Super exciting. Just to clarify, you were a participant in a study like this?
No, it was TMS.
Okay.
Yeah.
Sorry, I missed that connection.
No, it was it was like years ago. It was at the ICM. It was really cool.
I see. For those who may not have seen the chat, Mish actually shared the maps that are available on Neuroworld as a link. And questions, Lilith.
But I was the last one who raised the hand. But okay.
Junior's first.
Okay, thank you. So I have a question. Thank you, Chris, for first of all for presenting. So do they actually discuss, for example, ultrasound stimulation? It's one of the methods that's just started to be used in humans as well, and they claim that they stimulate deep structures. Can it go?
They don't talk about that.
But is it comparable, like how deep you can go inside the brain with that method? I don't know if you.
I mean, I don't know. In that case, they only try to target the hippocampus. So I don't know if there are some structures that would be much harder to reach. So I don't know if there is a limit to the depth, or maybe also maybe a resolution. I don't know the resolution of the ultrasound stimulation. I think also there might be a difference in the nature of the stimulation. I don't know how ultrasound is stimulating the neural activity.
Yeah, as far as I understand, in ultrasound is kind of mechanical, but this one is electrical. But and I didn't get exactly how did they manage to reach the deep brain. They used several coils that are directed to one focus point, or
yeah. So the like in the figure, they have two sets of electrodes. And the high frequency of the current, so if they were using only one electrode with this current, it would basically go through the brain without doing anything, because the frequency is too high to actually affect the brain, the cells. And they have two currents like that, and when the currents meet, they interfere, and basically the wave of the current, they subtract each other. So the frequency, so the speed, the number of waves per second reduces, and the current, so the nature of the current changes, and in that case, with a lower frequency, it is actually able to interact with the cells. And because the interference zone is localized, they can actually have a focal point in the area that they choose.
Okay, yeah.
So that's quite interesting. I was thinking a while ago that I thought it would be with a magnetic stimulation before. I don't know. I actually didn't read any other paper about this kind of stimulation, but I thought it would be easier to do with magnetic stimulation. I don't know if there are some studies about a method of magnetic stimulation. But yeah.
Yeah. Okay. Thank you.
Right, I sidelined Michelle and Hervé, so let's go in order of events. Michelle.
Thank you. Is that right? Am I understanding that right, that the stimulation actually decrease the activation in the hippocampus? If you look at the map, because so if you stimulate T11, then you have a comparable activation of the hippocampus with a sham, and if you stimulate T13, then this activation disappear.
Not
in the map, the other figure.
This I remember that they say that it increases the envelope modulation of the Bolt signal. So so I think that it increases the activity.
That is a counterintuitive result. That's why I was curious, and I wanted to make sure I really understood that right.
I understood that it increases it, but to be honest, I was not really focused on that. I don't know if they can.
It's an amazing paper. I mean, it's great. It's just like I was surprised to.
Because you would expect a decrease of the.
I would expect like stimulating to increase the activity.
I think it does. I think it does increase the activity. But I'm not sure how you can if you can modulate to reduce or increase. I guess it's more like a proof of concept. I don't know if you change the nature, the modulation of the current.
So Jan Fusak is actually online, and he's happy to represent this one. So go ahead, Jan.
Hello, everyone. Just first of all, can you hear me?
Yep.
Yeah. Okay. Thank you. I am currently working in a team which also works with DI. That would be Adam Williamson. I think he's one of the co-authors. I just wanted to clarify a few things. So when you were describing the actual stimulation at the focus, it's not that the frequency would change. It's that you get slow oscillations compared to the carrier frequency, slow oscillations of the envelope, right? You can also see it here, the red. You can see that the frequency is still the same. It's just
it's not the frequency that changes; it's the amplitude.
It's the amplitude which is modulated on the.
Yeah, I think I was confused with the slower envelope. Yeah.
So that's one thing. But in the end, it effectively acts as a slow frequency, slow compared to the carrier frequency, right? Which is something like 5 kHz or something like that. So you can get these like 10 or 5 Hertz amplitude modulations, which do the actual stimulation, because the neurons, they are not receptive to the fast frequency. And the second thing is the previous question was on the depth and focality, right? So compared with the ultrasound, here you have you can overlap more than just two fields, and that's basically carve out a more focal stimulation area, for example, right? In theory. So this has not this has been tested on
with the ultrasound. You can do that.
No, with this.
Oh, yeah, okay.
With the TI. So practically currently what we are doing, we are exploring the limits of how much in deep depth we can go and with how much precision.
Yeah, I guess it's just the beginning of the use of this technique, right?
Yeah. Also, what can be explored is different waveforms of the actual stimulus, right? Because here we have just classical sinusoidal envelope modulation, but one can play with different waveforms, different frequencies, and so on and so forth.
Do you know if it's already done with the more surface stimulations, like magnetic transcranial magnetic stimulation, or?
That's a really good question. I don't know. I don't know.
Sorry, I'm asking questions now.
The whole point to have a discussion, especially expert in the room.
I'm not an expert. That's all, as I said. Like that's completely out of my comfort zone. It was just a really interesting research.
John, did you actually have a question as well, or did you just want to chime in?
I just want to chime in to clarify the response from.
No, thank you very much.
Okay.
Ultrasound, yes.
In that case, Eri, thanks for waiting patiently.
Yes. Thanks, Chris, for the presentation. So I wanted to clarify or to see if I understood properly. So the protocol is you have a subject, because you need to localize the hippocampus. So you scan it first, after you go to the TI, and after you do the fMRI task. If it is correct, that brings me to my question. This is how long does the effect last between the stimulation and the fMRI, or even if we think about the treatment, how we can deal with the how long that lasts?
They have to target the target properly. I don't remember how they localize the target. But the effect actually in the second experiment where the stimulation actually affects the performance, they show that even so 30 minutes after, there is still an effect of the stimulation on the performance, but of course for long-lasting effects, because it was a one-time experiment. I think there need to be more studies about that. But I think the other types of stimulations show long-lasting effects when there is a repeating stimulation. So I guess if the nature of the stimulation is comparable, I would guess that the stimulation can have long-lasting effects if you do a protocol that is especially trying to do that.
Okay, makes sense.
Thank you very much. We're perfectly on time and I don't see any further questions. So let me remind you again that we have a CNS talk this Wednesday, 4 p.m. on the neurotransmitter receptor distribution and what that means for brain segregation, structure and functional by Nicola Palomero Gallagher. So do join us for that. And then the next Neuroccino on Monday will be by Eva. Exciting times. See you Wednesday or the very latest Monday next week. Have an exciting week. Bye.
See you.
Bye-bye.